Good afternoon, everyone, and welcome to a signature panel here in third annual Drug Delivery and Development Day, put together by Orientation Marketing and Fierce Events. I am Elliott Berger, a board director at Orientation Marketing, and I'm very happy here to host two of the most innovative company CEOs in biotechnology and bioprocessing on this great panel that talks about what's actionable now in bioprocessing in terms of a step change improvements in biomanufacturing for a step change in productivity, efficiency, and quality. With me today are Deniz Kent, a CEO of Prolific Machines. It's one of the coolest names of the company in the industry. And Himanshu Gadgil, we're actually here in the same room at DCAT. So welcome, Kent and Himanshu. Himanshu's CEO of Enzene, probably the pioneer and a leader in continuous biomanufacturing technology. So we're here to talk today about biotechnology and bioprocessing. And what I'm hearing and seeing in the industry, guys, is that it's really the current bioprocessing technology is coming to some limits in terms of addressing and development of the biomedicines of tomorrow. It's challenging, and the more complex the molecules become, bispecific, multi-specific, cytokines, fusion proteins, you name it. Deniz is the expert, all that stuff, not me. We'll talk about that in a minute. But it seems to be harder and harder to use existing bioprocessing technology to deliver what you need to deliver. It's also getting harder and harder to make these medicines affordable, and a subject that Himanshu cares deeply about. And it seems like a new generation of bioprocessing technology is really needed to deliver on that promise. So that's what we are here to discuss. Both companies have innovative, very different approaches to addressing these issues. So I'm going to ask Deniz and Himanshu to introduce themselves and their technologies briefly. And then we'll talk about the challenges and solutions and how that all works. So Himanshu, let's start with you since you're right next to me over here. Introduce yourself and Enzene, continuously innovative development and manufacturing organization that you lead.
Yeah, thanks, Elliott. It's a pleasure to be on this podcast with you, and really a pleasure to be here with Deniz too. I'm looking forward to also learning from the great things that Deniz is doing as well. So what we do at Enzene is we are one of the first movers in developing a continuous manufacturing process that's fully end-to-end connected. So a lot of time people talk about continuous, but what the way our platform differentiates is that it is fully connected. So starting from your upstream all the way to your pre-DS, the whole process is connected. And by doing that, what we are able to do is we are able to remove all of the whole tanks, which take up a lot of space in conventional manufacturing. And in a really, really small footprint, we can start getting up to 50, 60 kilograms of product, right? So in a 1,000-liter bioreactor, we can get up to 50 kilograms right now. And I'm, again, with some of the stuff that Deniz is doing, we can easily, with their cell lines, and our platform, we can probably reach that 100-kilogram benchmark. So, Elliott, as you were referring to earlier, right? The conventional paradigm of manufacturing in big stainless steel bioreactors, the industrial age manufacturing process where you manufacture the central big large location is changing. Products are changing. And what we are trying to do is really through bioprocess technology innovations, be part of that wave for changing the old paradigm of manufacturing.
Great. Do you want to also mention a couple of words about your personal background? I know you're a fascinating guy, and you really care about the affordability and the technology, not just because you have to lead this company. It's your baby.
Yeah. So I'll not say how many years I've been in bioprocess. I'll date myself, but I am a biochemist who's been working in bioprocess for many, many, many years. Worked for Amgen for a long time, where I was part of some fundamental work for defining QBD, CQAs. And after that, I moved back to India and then realized that cost of goods really starts mattering because patients have to pay out of pocket, right? So anytime you pay out of pocket, the whole cost dynamic changes. And with the socioeconomic state of where India is in terms of developing, I think cost and access and affordability are the key words for making sure that patients are able to get hold of these life-saving medicines, right? And that was the genesis of Enzene. So we were probably the 20th company who wanted to do biosimilar, and we did not want to follow the 19 other companies and make a product that is priced at the same price as theirs, right? So we wanted to change that dynamic. So yeah, I think my background, biochemist, worked in bioprocess for many years, initially developing novel biotherapeutics, and last many years, developing biosimilars, and now CDMO, where we are trying to, make our technology available to the world for their manufacturing needs.
Well, no, thank you for that, because I really wanted to make sure that that comes out. So I know you for a long time, and it really is important. I think best innovation comes out of necessity, and focusing on affordability, starting in emerging markets and biosimilars, but it's really now really also very important across the developed world, where the cost is really becoming a bit of a problem. Now, turning to Deniz, which is a bit of a different story. I like to think of Deniz as a California VC innovator, and even getting into bioprocess technologies in a different way. And when I try to follow Deniz, at least I understand what you're saying. When I try to follow Deniz, I have a much harder time. So Deniz, can you talk a little bit about yourself and your background and what led you to this really, really interesting field of optogenetics?
Sure. Thanks for having me. It's great to be here. So the problem that Prolific's trying to solve is taking biology from these black box systems that nobody can really understand or control, and reformatting it into a system that is very easily controllable and very easily understandable and optimizable by machines. And the way that we do that is by using light. And we've built this synthetic biology toolkit where any process inside of the cell can be controlled by different wavelengths of light. And you can layer on multiple different control levers orthogonally. And basically gives users complete control over cell biology for the first time. And that has profound implications for the pharmaceutical industry. We just broke the world record for the highest fed batch CHO titers ever seen. We're currently at 21 grams per liter in 15-day CHO cell fed batch, with only one optogenetic switch controlling one process, transcription. The ultimate goal is to have a CHO cell where you can control many different processes with many different colors. So, controlling transcription with one light color, and then, you could layer on metabolic control with another light color, layer on controlling the ER size and secretory capacity with another color. You can control receptor activation with another color. So we basically build the synthetic biology tools, the hardware and the software, to make this plug and play with your existing infrastructure. And so you don't need new CapEx spend, and you can just immediately 2 to 3x the productivity that you get from your current footprint, and have much higher margins on the stuff that you can already make. But the biggest benefit is in the stuff that you can't already make, or the stuff that you are trying to make, but the titers really suck. So the bispecifics or the trispecifics, the multispecifics. Currently, people just try and brute force it because we don't have a good way of doing it. So because we can't control the systems, people are just like, all right, we'll just have a ton of pools. And if we just have enough pools, then by chance, one of those pools might lead to a titer and quality profile that we want. But, at Prolific, we don't want to do that because it's not a very cost-efficient way of doing things. So what we have is a way to control the heavy chain and the light chain independently using different colors of light. So then you can precisely control the stoichiometry of these chains, and you can, instead of having hundreds of pools and brute forcing the process, you could just have, 4 pools and end up with the titers and the qualities that you want. So that means you can save over a month in cell line development timelines, and you can save a lot of money on your overall productivity going up and your downstream processing becoming much simpler because you don't have all the mispairings.
Well, now the audience can understand why a strategy and marketing guy like me has a hard time following Deniz's. If I didn't visit Deniz's facility and I didn't know it was real, I'd call them technology, which I think science fiction fans can understand. But it really works, and I've seen it.
Well, the scientists need to understand, Elliott, even if you don't
scientists will understand. Hopefully, the audience will understand better than I can. But what I get from that is control is good, and you get a much more efficient process that you can control the process. You can do it faster. You can do it with more control and more certainty. You can reduce the risk, and you can get world-beating titers for this bet. Now, you can also see why these guys are so passionate about their companies and technologies, because I asked them both to introduce themselves and their company, and they immediately got excited about their company. Didn't say anything about themselves, which is hard for a marketing guy to understand, but tells you who these guys are. So Deniz, before you go and tell us more about the technology, tell us a little bit about yourself and how you came to this optogenetics idea.
Sure. So I'm an immunologist by training, started my career working on CD8 T cells and how they get suppressed by tumors and why they get suppressed by tumors and if there's anything we can do about that. I got recruited into Big Pharma. I was developing a bispecific antibody for GSK, looking to induce allergen-specific tolerance. I then went into academia, did my master's and PhD in tissue regeneration, went really deep down the rabbit hole of how the liver regenerates, and was on the team that did the world's first single-cell RNA sequencing on the human fetal liver and on the adult and the human adult liver. Discovered a bunch of new cell types, spent half a decade characterizing one of those cell types. Generally just a science nerd, and yeah, really believe in the power of technology to move humanity forward and do useful things. I was first exposed to optogenetics while in academia, and one of my colleagues showed me a remote control mouse where it had a laser in its brain, and you could basically control a mouse in a maze using light shone in cells, and this blew my mind. I was looking at it with equal parts horror and fascination, and that was my first exposure to optogenetics. So these switches have been in the neuroscience community for about 15, 20 years. But more importantly, bacteria have been using them for billions of years. So really, this technology was not invented by humans. It was invented by bacteria. And that's why it also works so well. It's because there's been billions of years of R&D before humans came along to make sure these switches work really well. So we're just piggybacking off bacterial innovation. And yeah, I started Prolific 7 years ago, and recruited amazing co-founders and an incredible team, and we now run the best optogenetics R&D shop in the world. We don't want to be a CDMO. We want to be the best optogenetics R&D shop in the world in perpetuity. We want to build the best tools to control biology, and then we want to give those tools to CDMOs and to asset owners so that they can supercharge the amazing work that they're doing.
Well, now Himanshu, you get an idea why I call Deniz behind his back as Elon Musk of biology, right?
That's right.
I'm not sure if that's a compliment or an insult.
Once you get to 847 billion, we'll discuss it. So, guys, what I want to ask you, because that obviously both technologies are fascinating and different, but they are aiming at the same thing: make biomanufacturing of tomorrow more efficient and deliver the new medicine and a new set of molecules. And as I mentioned earlier, you go back, I don't know, 10 years, it was monoclonal antibodies and some more monoclonal antibodies and a couple of other things. Now you got bispecific, multispecific, cytokines, fusion proteins, this and that, and you name it. And the existing stuff is going to stay where it is, but the new stuff needs something new. So can you discuss a little bit about what sort of molecules and processes and technologies, medicines, do you think your technology is best suited for? Because there's no technology that's best suited for everything, and it's new approach is not needed for everything, but for some things they are particularly important. So Himanshu, what do you think about Enzene X and continuous processing and the savings and titers and efficiencies? Where are they most useful?
Yeah, so I think, as you were saying earlier, Elliott, I think the whole the field is moving as well, right? It's getting more personalized. So while before with brute force, we used to inject like 400, 500 mgs into a patient and then really the volume was very high, right? Now, medicine is getting precise. You don't need those 400, 500 milligrams. You just need micrograms of these newer products, and they work beautifully. Now, the problem is, as we start designing molecules where we take a domain from one molecule and stitch it with a domain from another molecule in the lab, it works great in the lab. But anything that is human engineered also has a lot of sensitivities in terms of either a proteolytic cleavage site or some aggregation propensity. And I've been in this field for many, many years. And what I've realized is the reason why MAB works is because it has survived millions of years of evolution. And anything that we create in the lab has not been exposed to that. So you start getting clips. So our very first application as a CDMO was with a company that had these multi-specific interleukins that were stitched together. They were manufacturing it in a 200-liter bioreactor. And the molecule was just getting chopped off by proteases that are there in the CHO cell. So, like 200 liters, 15 days, and they got only 3 grams. And so they came to us and say, okay, how do we solve this problem? Now, with continuous manufacturing, what happens is you're continuously taking the product out from this complex soup of matrix where you have proteases and all the bad stuff and taking it out into a more stable formulation, right? So they came to us and say, okay, we'll say, let's play with this platform. And in 20 liters, we were able to get 20, 30 grams, right, of this complex molecule. And remember what I said, they were only getting 3 grams in a 200 liter, and that too chopped off product. And when you saw our product, it was a single band. So I think the Enzene or continuous manufacturing platform naturally works better for these newer modalities that are highly precise, extremely potent, but also very often very unstable. And anything that is a recombinant extracellularly expressed, protein, you can use our platforms for that. So, but works beautifully for anything that's unstable.
Well, and then that's really important because I always say, look, there's so much risk with one molecule, and then there's efficacy, toxicity, cost, funding, priorities, and so on. The one thing that shouldn't kill a molecule is because we couldn't formulate and manufacture a promising medicine that people need. Anyways, on Deniz, in your shop, your co-founders named Max and Max, not even talking about Victor, were getting me confused with cytokines and fusion proteins. So same question to you, what is optogenetics really the best solution for?
Yeah. So it's the best solution for anyone who wants to deliver the lowest possible cost and the highest possible quality simultaneously, because there's no input into cells that give you cheaper control than light, because for light you need electricity, which is a rounding error, LEDs and circuit boards, which are both commoditized by consumer electronics. So it is basically as cheap as it gets for biological control. But interestingly, it's also as precise as it gets for biological control, because individual proteins inside of the cell can be dynamically switched on and off and tuned. And so any biological product can get made. But I'll give a couple examples of where control really matters. So if you wanted to get to, not just good MAB titers, but if you want to break world records like we have, then one thing that's really exciting about optogenetics is that it allows you to balance the metabolite pool with your production. So if you just have a constitutive system where it's constantly making the thing, then obviously before you even put the cells into the bioreactor, they're stressed because they've constantly been making it during the cell line development phase. And then once they're in the bioreactor and they're constantly making the thing, there's no focus for the cells. They're trying to grow up to max cell density, but at the same time, they're also trying to make a product. So with optogenetics, you can have them not be tired by the time they enter the bioreactor. And then in the first phase, you can hit them with no light or with a color of light that won't produce protein, but will speed up their growth. And then once they hit max cell density, you can introduce a transcriptional control lever that will then have a pulsatile production regime so that you can balance the metabolic pool with your production. So as the cells get exhausted, you withdraw the light, and then you can reintroduce and withdraw and reintroduce and withdraw. And it actually works It works in fed batch, but it would be even superpowered if you combined it with what Himanshu is doing and the continuous manufacturing. So it's compatible with whatever bioprocess method you want to run. That gets you to really high MAB titers. But just to give you some other examples, so with virus manufacturing, it's a sort of ideal control problem where the viral genes are toxic to the cells and they need to be expressed in a particular sequence in order to get the viral capsid assembled properly. And this is a temporal control problem. The way that people try and solve this today is you put the genes in transfection reagents, and then you add different transfection reagents at different times into your culture. But just like adding any molecule into a culture, you lose control as soon as you add the molecule into the culture because it goes everywhere. It's like dropping ingredients into a soup. The ingredients just go everywhere. So not all the cells get the same amount at the right time. You end up with an upset CFO because they see how expensive the transfection reagents are, and then an upset downstream processing team when they realize that 80% of the capsids are empty. So with optogenetics, one of the things that we've proven is that you can put all of the viral genes into a stable cell line where each viral gene is expressed by a different color of light, and then you can eliminate the biggest COGS driver with the transfection reagents, and then just shine different colors of light at the right time to get the viral genes expressed. You eliminate the empty capsid problem too, which reduces your COGS and also increases the safety. Another example, I already gave the stoichiometry of the heavy chain and the light chain example, but another example would be PTM tuning for biosimilars. So we've shown that we can control the enzymes that add the sugar moieties to the amino acids chain. We've shown that we can control those enzymes with light, so you can do precise PTM tuning for galactosylation, sialation, any of the sugar moieties you'd want to control. And we've demonstrated this, we've patented it. And we believe that it will usher in a new era of biosimilars, which we're calling bioidenticals. And there's a lot of excitement there because, the biggest cost for a biosimilar company is not manufacturing. The biggest cost is your clinical trial risk. And the reason why you have clinical trial risk is because your biological production method is messy and you can't completely match the PTM profile of the originator. You may get close, but not 100% match. And if you can 100% match, which is what Prolific can allow you to do, then your clinical trial risk can go to zero because you actually have completely replicated the originator, not just the amino acid chain, but all of the sugar moieties as well. That's another example.
A great examples. And I think, Himanshu will get excited about the bioidentical bit because that's even more affordable. But I think, what I was excited about is that while it's substantially cost efficient, more efficient and controlled and better quality for a number of things, it actually enables some of the classes of molecules that are really new and difficult that do not get yields sufficient to enter phase 3 and commercial manufacturing.
100%.
And that's really important to patients because you can then enable those medicines to actually exist.
Yeah, we're unstranding a bunch of stranded assets, especially the ones that are toxic for the cells, because you can hold them below a toxic dose by pulsing the light at exactly the intensity that you need to get to the titers that you need, but not so much that it kills the cells, which you can't do with a molecule, right? If you add in a molecule, you can't remove that molecule. If you want to dynamically change the amount of the molecule in a reactor, you can't do it. Same with a constitutive system. So light's the best way to solve that.
Okay, so when I'm also briefly, I want to touch on other things. On another thing that's important here is, like early development and cell line engineering and creating the right profile are very important. But all of that is for naught if you can't produce clinical material and eventually commercial material for that molecule to get through the stages. So I know both of you guys are working on that. I think Himanshu, you're a little bit further down that road. Can you talk about, how you manage not just the development process but the supply process?
Yeah, I think so. for biosimilars or for even anything that is novel, right? You need to have flexibility of scale because you don't know how much you're going to need when it gets commercial. Sometimes there's competition, so whatever your projections are for volume go out of the window. Suddenly there's competition. You don't need to make even a 20,000-liter bioreactor anymore, right? So what the newer platforms must do is they must offer more options for scale. And one of the things that continuous manufacturing does is there are three different ways in which you can scale. One is scale up, which is traditional. We go from 20 liters, which is equivalent to a 200-liter fed batch, to 1,000-liter, which is equivalent to a 15,000-liter fed batch seamlessly, right? Or you can scale on, which fed batch does not allow. So you can scale up like fed batch or scale on in time. So early on, you essentially you run your process for 20 days, and when you need more material, you start running it for 30 days and 40 days. And just like Deniz was talking about a world record in terms of titers, we kind of have our own world record in terms of reaching 100 grams per liter with cell lines that are almost half of what Deniz has, right? So if we combine these together, I think we're going to have a super duper world record. But again, scale on is an option. And the third option, which I'm most excited about, is scale out, because the platform is very capital efficient. You don't need to invest $250 million to be able to manufacture 100 kilograms per month. You can invest $50 million, lot more compact footprint, which means you can easily scale out. So we've been doing geographical scale out. So we were in India, now we are in the US, but we are also looking at this hub and spoke model to put it wherever the manufacturing needs are. And unfortunately, the world is changing again. I mean, the world is always changing, and now we are changing more towards from being a flat world to, again, some kind of a concave, convex world where, everybody has to start looking at taking care of their own manufacturing needs, right? Especially post-COVID, I think that has accelerated where there is a lot more push for localized manufacturing, right? And what this platform allows us to do is we can put it in a very small room somewhere in Hawaii or at the same time in Siberia and still make it work, because you don't need a lot of human capital. It's very automated. You don't need a lot of space and you don't need a lot of capital, which means we can start localizing manufacturing with this platform.
That's right, but I'll go to Hawaii, go to Siberia.
I don't know.
So, okay, briefly, Deniz, we have a couple of minutes. I know you guys also have given a lot of thought on doing that. And I saw some really clever prototypes in your lab. And I know you've been experimenting with using your technology to basically attach it to any of the existing sort of bioreactors. You don't have to replicate the technology and allow clinical and commercial production using optogenetics for both in-house with pharma companies or with the chosen CDMO. Talk to us for a couple of minutes about that.
Yeah. So, like I said at the beginning, our goal is not to be a pharma company or to be a CDMO. Our goal is to invent the best tools in the world to control cells and then to make them available to everybody who wants to control cells at a lower price and with a higher degree of precision. So any pharma company can come to us, and if they want to do in-house manufacturing, we can set them up to do that. If they want to go to a CDMO, we can set them up to do that. They can either choose the CDMO that they want, or if they don't want to do that, we can point them towards CDMOs that we're already working with. And yeah, we can be flexible.
Flexibility is really important because, some pharma companies prefer to do stuff in-house, especially with priority molecules, but there's a lot of virtual biotechs that have very promising molecules and are really well suited for this. But, thank you guys for a great discussion. I think there are two very fascinating technologies that are both actionable and real now. I think they both can help deliver not only breakthrough cost efficiency, but also help the more complex molecules actually make it to the clinic and make it to the patient, which is what we're all here for. So thanks for joining. I would encourage everyone to contact Enzene and Himanshu or Deniz at Prolific and pursue this further, because I believe that any technology that kind of is shown to make stuff more affordable and to bring more medicines to the patients is really worth doing. Thank you very much for joining, and stay tuned for the rest of the program.